So, one thing, you realize that I don't allow you to fall asleep, so you never know who's going to be the next one to be asked the question. First thing, I realized that it's pretty frustrating if you stand up there and everybody's just twittering all the time. I mean, it's like in the subway. People don't look at each other anymore. So, in the interest of the speakers and myself, you may use social media, Twitter, as much as you can. That's good for NextLevel Pharma. But sometimes just concentrate also on the talk, because you might be the next one to be asked a question. Oh, yeah. So you might think this vision is far away, right? The fridge is going to tell you what to eat. This vision is actually a result of a discussion that I had with a senior health information technologist at GE when we discussed how we are going to collect data and what we're going to do with this data. So eventually, in the future, we will all have our molecular profiles, and these will be entered in some kind of database, and it will help us to manage our health. We go to prevention, and we will know our bodies a lot better. Interestingly enough, just about 4 weeks ago, you know all about Watson, what Watson is? IBM was investing about 80 million in pathway genomics. Surprisingly, the CEO of pathway genomics is on the board of the Watson team. So that's how things work out. And they were advertising that they will bring up an app that helps you to manage your nutrition lifestyle. I mean, this is a genomics company. I don't know how far they want to get there, but it's very ambitious, and it shows the trend. It shows who's going to be the new players. So now I'm going to advertise for a company that is actually not incorporated, but that doesn't mean we are not working. We're a little bit a strange bunch of people, serial entrepreneurs, number crunchers. Some people are still at senior executive persons at companies, pharma companies, and we're building a solution that helps to solve some of the problems Patrice was addressing. We're not jumping in the same space as Calico and human longevity is, but we feel that inflammation is a fundamental process affecting most conditions, and it's poorly understood. And with all these multi-omics technologies, it's actually possible to decipher inflammation much better, and that's basically what we want to do. Just talking about how good or bad health is today and how patient-centric drugs and how effective they are. Just imagine your wife is diagnosed with like 35 with rheumatoid arthritis. What will happen? She gets the cheap drug first, right? Because that's the healthcare plan. And there is a window of opportunity where we can drive the disease into remission. So the follow-up costs for society would be rather low. We can really push the disease almost in remission with the very expensive drugs. The problem is the expensive drugs work for maybe 25, 10% of the patients who just don't know. Right, Bill? Big problem. And that's why the payers are reluctant to allow us to use the best drugs for the patients very early on. That's a problem so far not solved. But the socioeconomic burden is tremendous. Good morning, Herr Weber. He's one of my colleagues. An engineer, by the way. He told me that medicine is not a science, and he's right. So, that's how we diagnose disease today. It's like these little puzzles for kids, right? It's like a ridiculous 10-digit score, like how long do you disease, how many joints. There was a recent review, a disease formerly called rheumatoid arthritis. I like the title a lot, because it's most likely if drugs would work in all patients, we would have one disease. But since drugs don't work in the same disease, most likely we have many diseases that for whatever reason affect the joints. So the idea must be that we better stratify these conditions that somehow look alike, and we will use all these kind of multi-omics technologies in the future. And how much is this in a cost perspective? I'm not going to go into details of the slides, but we calculated how much we could actually save if the test would be so good that we just eventually would be able to diagnose 50% of the patients correctly. the annual savings would already be 900 millions if we would be able to target 80% of the patients correctly. We would save 1.6 billion a year. And the revenues, if these kind of tests corresponding to these tests, is like $1 billion US. So the market cap is actually not that bad. It's very interesting for payers. And one thing that Patrice said, well, science should be more patient-centric. I think we also have to mobilize the patients to ask for what they want, because breaking up the silos needs breaking up the FDA, breaking up whatever. It's like mobilizing people to eventually change and push payers to also get engaged in bringing these products to the market. So the idea is basically to reduce the cumbersome investments. I mean, we all know that the R&D model in pharma is broken. It's not sustainable. Up to 5 billion are the last numbers that I hear to bring to an NCE to the market. The studies are just obscenely expensive. I remember talking to Novartis. They were moving out of inflammation for some time because they said, well, in rheumatoid arthritis, we just can't sustain the requirements anymore. It's not financeable. So we need a new paradigm. And precision medicine is most likely focusing on individuals, like on also including rare diseases as role models, and identifying the responsive patients very early on, so you can reduce, possibly with adaptive clinical trials, the cost significantly and also spare a lot of time. So what we are going to do is breaking up silos, and obviously we can't do that alone. This is a complex slide. I just wanted to point out that it takes, of course, a lot of players, like Patrice is setting up his network. We are also setting up a network. The core competence is basically number crunching, algorithms, and I'm going to relay on this topic a little bit more in detail, collection of samples in a standardized manner, because that's very crucial. So, who of you has heard about precision medicine in this audience? Raise your hands. That's pretty much, actually. I'm surprised, but most of you are foreigners, right? Who has heard about precision medicine and is from Europe? Less. So that shows a trend. There are a lot of people that are aware of this change of taxonomy, which was actually introduced 2008 by Clayton Christensen in his book, The Innovator's Dilemma in Economist. And it's been adopted by the National Research Council in the US, and meanwhile by Pfizer. Some of you know that I'm somewhat a LinkedIn wizard, so I posted a video from Pfizer where they explain what precision medicine means to them just a couple days ago. So the idea is going away from what medicine is in a general practice, particularly in the rural areas where you have just an MD, you have fever, you have cough, your kids go to school. So he diagnoses you by this kind of context. If he doesn't ask you where you've been traveling, maybe you've been to Liberia before, that's a big problem nowadays. But basically, it's a very symptom-intuitive medicine which is being practiced all along. If you look at more clinical-orientated and R&D-oriented medicine, it's based on the evidence from large cohorts, right? We look how much, if we basically talk about rheumatoid arthritis, we compare oranges, apples, and we treat them all with the same ingredient, and then we look what's actually killing the germs best. So that's this cohort-based analysis. And what Clayton Christensen was predicting, and we know Kosler is one of his biggest fans, is that we in the future will have a more precise medicine. I never liked personalized medicine, by the way, because medicine is always personal. It was personal and will be personal in the future. And if you have a physical exam, you know exactly what I'm talking about. So I'm not talking about prostate. Yeah. So that's digital health, you know? No, just kidding. So generally speaking, we will have more sophisticated methods to stratify patients in the future. But eventually, with the convergence of mobile technology, this will help us to diagnose disease precisely and even become preventative in a real-time everywhere in the world. And it's not that far away. There are big visionaries like Dr. Xiong. I can't spell this name. You probably read it it's Nanthealth, if you want to Google it. He's building up these ecosystems with all his money that he made as an entrepreneur. So, Patrice was showing the slide. Where all these data that we are going to integrate coming from? It's this kind of multi-omics. Omics is nothing else but mass data from exposome, microbiome, metabolome, proteome, transcriptome, socionome, whatever. It's going to be integrated, and it's somewhere an Orwellian vision that we will have all data collected and we will have our digital molecular fingerprint. But at the same time, it's like Google Maps. We will have to we can navigate with our health around, but the trick is we have to create these maps. And again, eventually in the future, I'm going to skip this slide, we will all have our personal molecular profile. We will have it very early on and will accompany us throughout life. One thing that actually was striking my mind and very inspiring was the paper of Mike Snyder. Who's familiar with the Snyderome? Not too many, so I have to explain a little bit what he did. Mike Snyder was collecting about 20 snapshots over 18 months, 20 time series measurements of his blood, multi-omics, and generated masses of data. And with this approach, basically, he could decipher when he got the diabetes, and it was correlated to some kind of viral infection. He could also demonstrate that our intra-individual variations during various days is so high that it doesn't really make sense to do clinical trials as we do it today with the very sensitive methods, because we have too much background noise that we create with our variations already. So the idea would be in the future that we go away from cohort analysis, but do intra-individual longitudinal analysis. And that's actually what you could do excellently in chronic diseases, because these patients get different drugs anyways. You just have to monitor these individually and compare them to themselves as their own control. That's part of the idea we are pursuing. So this is again the Snyderome. Well, why I'm bringing up this slide is 50 terabyte of data. That's pretty massive. And it's actually, if you try to download that from their server, talking about cloud, it takes 2 weeks. So that's not very fast, and you cannot process that. So that's not a business model. And the costs just for this individual is not scalable. But working with engineers that have been bringing several products to the market, you can look into this model and figure out where are the breaking points where you can actually make that a business model, make it a model that you can apply to the clinic. So I always show this slide because of the uncork. That means apparently scientists are always drunk when they have good ideas, to some extent. But it's also obviously we're dealing with something that we're not trained for. It's a massive problem. And another problem when you talk to MDs, I mean, they learn statistics, right, at med school. It's like, okay, we know what a t-test is, and we think somewhat linear. But this is basically what happens with an immune reaction. It's a chaotic system. It's a dynamic system. And we monitor it in vitro with like 2 or 3 time series and do a linear testing system. I don't know whether this is appropriate for the methods and times and mathematics we have today. Next thing is, actually, I want to show you a video, which I find rather impressive. Hopefully it works. How can I do that? Here, there it is. Oh, yeah. This is a simulate. Okay. Forget. Just go back to the presentation. It's a kid's toy. It's like a pendulum. It's a triple pendulum. And if you have 3 pendula integrated in one, it behaves chaotic. And that's very impressive because it's just, you think it should swing like a normal pendulum, regular, but it absolutely doesn't. It's just 3 factors. What means yeah? Oh yeah, thank you. So if you measure 3 interdependent, because that's what we talk about in biology, parameters, you get these kind of time series. I mean, what are we doing? Right? That's something, and I saw that there is a toy that you can buy for kids in a store where the kids were playing with that, and that was like 15 years ago, and I thought like, what the heck am I doing? This is a chaos pendulum. So 3 interdependent factors. So let's go back to the presentation. I hope this is impressive enough to make you think. That's the basic idea. So, talking about how does nature do things? Nature has actually trained us to use our senses by pattern recognition. Ancient sailors were able to navigate 3,000 years ago by looking at the stars. They were pattern recognition. And all our senses use pattern recognition. The immune system, if it would be rigid and not dynamic and flexible, we would just all die because the bugs are very flexible. It's a very archaic principle to recognize irritations in nature. I like this slide because it's the battle between the cuckoo and the bird that is hosting the cuckoo's egg. And actually with pattern recognition and pattern discovery, we can figure out, meanwhile, that this is a battle that is ongoing. I mean, the cuckoo tries to mimic the patterns that we just don't see. Do you see any pattern there? No. A machine does. But what I want to demonstrate is there are patterns in nature all the time, and they're very dynamic. But if we look at disease as this battle between health, disease, these are patterns that are shifting from one state of aggregation to the other one. And we have the tools. I mean, face recognition, for example, developed by Facebook. It's very dynamic. It can actually recognize faces during different lifestyles, during different stages of, let's say, evolution by surgeons. As you can see, Sylvester Stallone is still being recognized by this algorithm. And that's a dynamic way of recognizing patterns. Similar thing. All of you will instantly recognize what's wrong with these slides, or when they've been made, right? Some of them were made before 9/11. And it doesn't even need a baseline. Very important. How many biomarkers do really have a reliable baseline? How many do you think have been really, there's a baseline and against this one we can measure? It's like 11, not more. Everything else is very dynamic. And when you talk about how different we are, you know how we'll appreciate how difficult it is actually to find biomarkers that you can measure against the baseline. It just can't work. So, patterns. Patterns might be dynamic. They might accompany us as a fingerprint in our life, and they may evolve with us, being specific for our own metabolism or based on our genetic fingerprint and the combination of the exposure to our life environment. Talking about tools that are available, we do not have to reinvent the wheel in pharma. We can use, this is a slide, actually this guy is still alive. He's been analyzing the interaction in a criminal network by doing this kind of mobile communication analysis. And I mean, mafia is a disease, right? So we can actually map other diseases as well with the same tools. So they are available. We just have to apply them, and that's what has been done. A lot of people try to do these things. This is a study that I love actually, because it's very much in line with what we want to do. It's the University Hospital in Toronto, and there is a condition in children, pediatric arthritis. And from our clinical perspective, we would say there are 6 conditions However, if we just look at 50 parameters and use machine learning, we can actually cluster the condition, this condition in 11 molecular clusters. So do we know what that means? No, obviously not. But if we would start monitoring these kids over an extended period of time while they take the drug, we will see how these clusters respond to drug treatment, to change of behavior, change of nutrition, whatever. It's going to give us much more and better insight than we have by our, let's say, old, archaic, clinical way of diagnosing disease with these kind of scorecards. So machine learning is a powerful tool that's going to help us stratify patients even better. And these are data, it's purposely unsharp, so you're not going to be distracted to the message that I want to convey, which is we got data, metabolomics data, from one of our partners, Metabolomic Discoveries. This company is actually represented by this young lady here. If you want to talk to her, she's the business development head. They gave us the data from 1,200 patients. 132 patients, 1,200 metabolites. And what's the difference is their number crunches took about 2 days to stratify the patients in several clusters, to develop a model. We used the software from the CIA. You can license that. DARPA actually also offers these kind of tools, so you just have to go there and ask. And we have this guy that always tells me, I developed the software, so when are the Taliban striking next in Afghanistan? So disease is also some kind of a terroristic attack. What we did is we had, of course, to adjust the data to some extent, but it takes 1 minute to analyze that. And that's very important, because if you want to scale omics, the most cost-intensive part is not creating the data today. We can scan ourselves for 1,000 euro. The genomics revolution is like, it's way below full next-generation sequencing is $1,000. We can generate the data, but analyzing them is exponentially cost-intensive, and we have to automatize this process. But I love this effect. Don't you like it? Bong. It's like woof. So, garbage in, garbage out. There will be a study coming out from the Helmholtz Center in Munich. Jurek Adamski is an advisor to Metabolon as well, and he's one of the gurus in metabolomics. And they've been looking at the quality of samples and how blood collecting is affecting the quality of analysis. So how do MDs take blood? So they would take it, put it in their pocket here, run around, chat with the nurses, and eventually after 1 or 2 hours, it will arrive in the lab. And since this is depending, it's wintertime, summertime, temperature, do you really believe with a highly sensitive method like mass spec metabolomics, this is the way you should collect blood? Not. So there was, for example, a large IMI project, 15 years blood collecting, etc. They started analyzing. After 400 samples, they stopped because they figured it's garbage in, garbage out. Next thing, if 80% of published data are not reproducible, not reliable, not valid, it's a nice business model if somebody comes to a, let's say, analytics company, not to name anything, in Silicon Valley, well-funded, and tells you, extract anything you find in the literature. They will do it. And you pay a lot. But what is the content that you get out when we know firsthand that 80% is crap? So Pharma has a problem because we cannot rely on the science academia is providing to us. And so the idea is what we have to do, and I think that's the industry that has to do, is we have to create SOPs, and that starts with simple procedures like collecting blood. If we want to have more outcome-based, better data that help us to stratify patients, if we want to have new ideas and also apply these multi-omics, super sensitive methods in a value-generating procedure, we have to create SOPs. And I'd like to all read this sentence very careful. I'd love to be the devil's advocate, but this is coming from the National Biomarker Development Alliance this year. How many academics are in the room? None. Good. Yeah, because, I mean, I discussed it recently at the European biomarker event in Munich, which is driven, it's an academic cluster. And if you tell them, look, guys, you're MDs, but we're producing crap, that you don't get friends there. So, generally speaking, but at the same time, I mean, the idea of this slide is, this is a difficult task. So, you can go out and tell your postdoc, so you develop SOPs for me. Is this going to work? No. So the industry has to develop these SOPs. There has to be awareness that we need to create an environment where we start sample collect to create SOPs for all these multi-omics technologies in a way that make data comparable, repeatable, and so forth. And there are two publications that I'd like to recommend. The National Biomarker Development Alliance just a week ago published a new review. There were two these years. One is the earlier one in September. That's where this quote comes from. And there's also a very interesting publication talking about breaking up silos from the Manhattan Institute by Peter Huber, because he criticizes R&D, FDA research paradigm, which is 60 years old, in the context that we also have to break up the silos there, and also addresses the problem of standardized data collection. So, and to summarize my talk, there's a company that's trying to put all these pieces together in a vertically integrated value chain, and we're still open for collaborations, funding, lots of money. So, and I run the group Precision Medicine and Big Data on LinkedIn. So you're happy to join. Thank you. Am I on time? Good. So.
Yeah. 1 second.
Oh, many questions.
So I thoroughly agree that we need to look at diseases from a molecular perspective and diagnose them at a molecular perspective, because looking at a gross phenotype and the measures that we have, like you pointed out for rheumatoid arthritis, don't pull out the nuances of the disease for each individual. But it's a real challenge to shift the provider space over to that, because that's a huge change for the medical doctor training, as well as how healthcare is actually delivered. And we still don't have a good molecular basis for the different diseases. What you pointed out for juvenile arthritis is an excellent start. How do you see getting from where we are, which is, I agree, somewhat archaic?
Well, it's actually pretty cumbersome, and we're really pushing the limits in our fundraising process. But the thing is, if pharma is not engaging, you see companies like Calico, Human Longevity. These guys are exactly pursuing the same business model. I mean, 23andMe's business model is all but democratizing the knowledge about your genomic background. It's about getting the data of a whole family with some questionnaires and then having their genetic profiles, and they do deals with Pfizer. So the one that's really, the business model of the future are the one that has the data, the reference databases, and the algorithms will win. That's like Amazon, right? Personalized services. They know you. The better they know you, the better are the suggestions. And that's where a lot of new players come to town. It's like IBM. So pharma is really being challenged actually to go into this space and be innovative, because otherwise they are already under pressure. There was a recent article, when will Illumina buy Roche? Because the power is shifting, right? The patient wants the right drug at the right time, and we empower the patient in the future. They are empowered by the social media. And the FDA supports that. So eventually it will be like the diagnostics will decide which drug comes to the market. KOL investments will become obsolete in the future, unfortunately. So.
I have a follow-up question regarding the previous discussion around patient privacy, and in the context of the example you gave where we have now in the public domain an extremely well-characterized individual in terms of the full set and the implications for that. Doesn't it mean that actually that at some point in the future people will be able to choose the level of privacy they want? So in the Schneider example, we have a great deal of non-anonymized, personally identifiable omics scale data.
This data safety, security, privacy is a topic for 2 or 3 sessions and for several days. But I personally have this kind of thing. A lot of things change, and people may have a different opinion. It's a cultural thing. Like 50 years ago, when we're living in a small village in the mountains, the grocer knew everything about everybody, right? So there was no privacy. Now we live in the big cities. There's a lot of privacy, but our kids give up privacy in Facebook, and now they learn. Oops, I didn't want to do that. I think this is a very dynamic process. What I find much more interesting is, will our data become somewhat a currency? You know, like to whom am I selling my data, or whom do I allow it to access the information about myself? To what extent?
And so the idea of these kind of micropayments, I guess, is what you're saying here, that actually the information that we have ourselves, if someone else is going to use it, we should have some kind of micropayment system depending on the amount of data and the value that it has.
Right. And at the same time, we talk about a major debilitating disease in rheumatoid arthritis. I think a lot of patients would be willing to give up a lot of data just to be in the state of remission and remain in the state of remission. So it's a matter what we are going to do with these data. So, Patrice?
Just to add very quickly. It's also creating an intergenerational issue, because if you give up your DNA information, you engage responsibility over your generations. So kids are now complaining about parents releasing DNA information.
Right.
So one more question there. So Daniel Ziemek with Pfizer, and you mentioned Pfizer a couple of times, so I thought I should speak up. I guess I was wondering about, you talked about precision medicine, and I agree, I think that's an important piece. And you also
Morten Sogard is a friend of mine.
Oh, I see, yeah. And I guess then you criticize, or at least you call into question the quality that academics generate these biomarker data. But my sense, precision medicine, at least as we usually understand it at Pfizer, the idea is to look at drug response, right? To find the patients who respond well to a specific drug. And I think some of the examples you gave are more on disease heterogeneity, like your rheumatoid arthritis plot there about how the different, like in a disease population, how that would be different molecularly. And I think they're related. But I was wondering if you want to comment a little bit more on how you see your company or the vision for precision medicine more in the drug response space, which I think is a little different from the disease heterogeneity space.
I think it's not that much difference, because every drug will have its own signature as well. And that's actually what makes it so appealing. Because when you measure everything, you will have both. You will have the baseline, you will know when the patient is in remission, and there will be some kind of patterns. And we know already, for example, with urine metabolomics, you can differentiate if the patient got infliximab or Imbrel. I think that's pretty impressive.
Right, but at least maybe I would be interested if you could point me to examples. So first of all, I think lots of the drug response data is generated in clinical trials, which at least I hope are better controlled than the ones you're criticizing.
Right.
At least I would say the ones I've looked at internally. And so that's one point. And the others, I was wondering whether you can give examples where it actually worked, right? So where you're specific, I've seen many data, infliximab, Enbrel, all that. You can differentiate, but can you predict who's going to respond to something? I was just wondering, do you know examples where...
No, we want to do it.
So
that's like, it's how we have to put the pieces together. There is a lot of evidence, actually, that you can predict certain things. Actually, maybe Bill, he's working in this field, could comment.
Actually, Jennifer can probably talk to this better than I can, but from blood transcriptomic data, we can actually predict whether an anti-TNF is going to work for a rheumatoid arthritis patient or not, to a very high level for a subset of that population. With more data, we could probably do a lot better. But disease drug response, the drug is attacking a particular mechanism. Once you identify the different mechanisms that are driving disease in a particular patient, which there are many different molecular mechanisms that are driving rheumatoid arthritis, and it's purely matching that mechanism of action from the drug to the disease-driving mechanisms. So I agree with Thomas. They're exactly linked together.
I mean, currently, when you look at, this is a very specific topic, but of priority to me, apologies. We also have these kind of silos how you approach biomarker discovery. If you do it agnostic and you look at everything, that's going to give you a different picture. Immunologists will look at proteins and cytokines. They are unspecific because they're part of inflammation. Do you really expect that they respond differently in different conditions? I did animal studies with endotoxin. There are responders with 10,000-fold responses in one cytokine, and some don't respond. So it's just basically we have to look at the whole picture. We can take these analogies where machine learning pattern discovery is really very powerful in finding very hidden data that we just can't see. The problem is we have to go off our hypothesis-driven. That's very hard for us. It's not bad science not to have a hypothesis. In fact, it's even more challenging to make sense of the data and validate it afterwards. That's what I tried to explain with the slide on juvenile arthritis. We just don't know what it means. But it tells us something different which we couldn't see before. So I think we should go on, right? Nobody's taking the time, but one last question.
Yeah, a general remark. I mean, we have been hearing a lot about confidentiality, patients and everything, but how many in the room are really talking to patient organizations? Because if you're talking with chronic diseases of any kind, I mean, these people are extremely willing to give any data set available. So I think we clearly have to use them more. They are huge organizations within Europe who are organizing this.
Yeah, if I may, another comment on what the person from Pfizer was saying. Actually, I believe nowadays there's a shift in the drug-related information. The data which is attached to drugs already on the market are growing and far exceeding the data that we collect in our clinical trials. Okay. Therefore, the value proposition is being revisited by payers. What we believe was a very strong value proposition coming from our clinical data are going to be accepted for a while, but they will be challenged again and again and again. And we are getting into a world where, let's say, being able to sustain a product on market for many years is going to be much more difficult.